This is my neural cell, and this is my identification card. Let me just film the body and the labeling. So this blue part right here, those are the dendrites, and the dendrites receive signal from other neurons so an action potential can occur. And this right here, those are the white pieces. Those white pieces are the Schwann cells, and these are the cells that allow the nerve impulse to travel along the axon so that the action potential can happen. Each one is individually wrapped with myelin sheaths, and those myelin sheaths that wrap those Schwann cells well, their purpose is to protect them so that none of the cells are damaged and the nerve impulse can carry on. In between each of those Schwann cells, the nodes of Rivera are present, and those cells allow the process of nerve impulses going through the neuron to be faster, and so none of the impulses get stuck in the process, so everything can go on smoothly. And to move forward to the axon terminal, which has tiny neurotransmitters at the end of it, and those at the end of it are chemical messengers that allows the generating of the action potential. Okay, first we have a resting membrane potential. It is at 70 millivolts, so nothing is happening to this neuron, and it's waiting to be stimulated by an external source. First, we have potassium ions, which are right here along the neural cell, and those potassium ions are located in the intercellular fluid, which are negative, and the sodium ions, which are right here, are located in the extracellular fluid. In order for a reaction to happen, a threshold must hit with 55 millivolts. When this occurs, the sodium ions influx into the cell. So this is the process right here, and then it breaks the visceral, and the neurotransmitters go to the dendrite right here, and that's where the process starts. So when the hit occurs with the 55 millivolts, when this occurs, the sodium ions influx into the cell and into the intercellular fluid through their voltage-gated sodium ion channels. This occurs, changing the permeability inside from negative to positive. And this is known as depolarization. Next, once this happens, the sodium voltage gap channel must close and the potassium voltage gated ion channels open and allows potassium to influx. So potassium is gonna influx and then sodium is gonna outflux. So they're pretty much changing places. And they switch positions. So you have a sodium ion which moves from extracellular into the intercellular fluid. Then the potassium ion moves from the intercellular fluid to the extracellular fluid. Once that happens, the potential action can occur. It travels along the axon. So it goes right here and travels along the axon. It goes right here and travels along the axon right here. And then once that happens, the potential action can occur. And then it goes to the axon terminal, which prepare the neurotransmitters to do the action potential. And then this is how the whole process happens. So after the action potential happens, that's when you get the sensation of like getting hurt or like getting burnt and stuff like that, and like the feelings that you feel. So